Hi, so thank you. I'd like to thank the organising committee for inviting me to give this talk today. This will be the first talk of this type that I've given, so I do apologise if it's still a little bit too scientific, okay? So what I'd like to do today is just give a brief overview of some of the scientific research that has been happening in the field of MND for the last few years and what this really means for the future. So where we currently stand is that we know that since the initial demonstration of this modest improvement in survival in response to riluzole in MND patients, we haven't really progressed so far in being able to develop new treatments for this disease. And this is really because we haven't been able to identify any one trigger that sets this disease off or any one real cause that is amenable to treatment. And, you know, a lot of the reason for why we've also not been too successful in translating what we see in animal models into the patients is that unlike the animal models where we can control the onset of the symptoms and we can control where these symptoms arise, MND is very heterogeneous. And this is best exemplified in the clinic. And what you see is that the symptoms can develop in different areas in these patients, with some patients developing symptoms in the bulbar region, in their upper limbs, or in their hind limbs. Now, what's more is that MND can develop at any time. And while most people develop MND between the ages of 40 to 60, you do see patients who are 80 and some who are as young as 20. And finally, the rate of progression is very different between patients. And it was in 2002 that Shyuan and colleagues demonstrated that the rate of symptom progression early in the disease process can really predict the outcome, in that patients who had very slow progressing disease had a better percentage of survival compared to patients who had very rapid progressing disease. Now, despite all of this clinical heterogeneity that you see in patients, what we do know is that the pathological manifestations are generally the same. So first, what we see is the loss of these cells in the brain that are associated with controlling movement. And we can see here in this image in red, I'm not sure if you can see it too well, these are the cells, and they're in a very distinct area in the brain, in the motor cortex. And what we can do is when we use animal models of motor neuron disease, you can see that in a normal animal, there are quite a lot of these cells in this area of the brain. And when you look at animal models that have developed the disease, you see the loss of these cells. Not only do we see the loss of cells in the brain, we also see the loss of cells in the spinal cord. Now, these are the motor neurons that send these projections out to the muscle, and they basically relay the information from the brain out to the muscle to control movement. And we can see that again in animal models. With animal models, in a normal animal, you see quite a number of these large cells in the region of the spinal cord, whereas in animal models and mice that are of the same age that have the disease symptoms, you see the loss of these beautiful cells. Now, when you get the loss of these cells in the spinal cord, you're of course going to get the concurrent loss of the connections that it makes out in the periphery and the skeletal muscle. And this is called the neuromuscular junction, which Rob kind of alluded to this morning. And what happens normally is what you get up the top in a normal individual or mouse, you get these nice long projections coming out from the spinal cord. And we've labeled these here with the green marker. And what these projections do is they make contact with an area of the muscle that contain these proteins, which we've labeled with red. And these proteins basically respond to the chemicals that are released from these projections, and this drives muscle contraction. And what you can see in an animal model where you see the symptoms in the hind limb or in the muscle, you get this retraction of this projection. So the green is starting to move away from the muscle, the projection is moving away from the muscle, and this basically doesn't allow for that communication to really happen. And this really underlies a lot of the muscle weakness that we see in the disease and the eventual paralysis. So with scientists being armed with the knowledge that these structures, these cells and these muscle connections are really affected in disease, we've come up with a few hypotheses as to why these structures are susceptible to death. And so how far have we really come in understanding what causes the death of these structures in MND? And I'll just start off with genetics, because since the initial discovery of the SOD gene, there was a bit of hiatus in the gene discovery side of MND research. And with advancing technology and the ability to sequence the gene really, really quickly and at a relatively affordable price when you think about scientific technology in the whole scheme of things, of the 114 listed MND genes or associated genes that are on this genetics database, you get the usual suspects coming up. And these are the ones that people generally talk about when they're doing MND research. And they are the TDP43, FOS, C9orf72 genes, and so on. And what we can see from this diagram, which Renton sort of published very early this year, is where we are with the state of play, is that a lot of these usual suspects account for about 70% of these familial cases of motor neuron disease. And these genes also account for about 10% of the sporadic cases of MND. But what you really have to take in is that despite all of this progress that we're making in genetics research, there is still 30% of unknown causes here and 90% here. So this really leaves us with a lot of room to really discover new genes that might be associated with MND, and it's probably likely that this will end up being a genetic disease. Now, the contribution of genetic mutations to MND can only really be demonstrated when you consider the function of the protein that arises from that gene. And so what a lot of scientists have done is they've looked at the consequences of these genetic mutations and what happens inside the cell. So if we start with SOD1, which was the first gene, it's really important the protein is really important in modulating oxidative stress inside the cell. And for all proteins to function properly, they have to come together in a very specific conformation. And if the proteins don't fold in this particular way, they basically don't work. And so the SOD gene really causes for some of these SOD proteins to misfold. And other proteins that are associated with MND, like the TDP43 and FUS, they have these special regions in their protein sequence which actually makes them susceptible to misfolding anyway. And so when you get these misfolded genes, and you also have mutations in genes that are involved in basically degrading these proteins, and a lot of people are going to use terms like the endosome and the ubiquitin proteome system and autophagy. All of these terms really refer to the ability to take protein that's not working and shouldn't be there and to get rid of it. And in MND, what you see is a lot of these functional pathways are mutated, and so they don't function properly. And so you get the buildup and aggregation of these misfolded proteins, and this is eventually toxic to the cell. Other mutations, like C9orf72, which is also involved in this degradation pathway, has also been suggested to control RNA transport and RNA function. And when RNAs are not turned into proteins properly, they get packaged into these little granules. And so when these proteins aren't made, you also get the loss of protein function. Now, profilin was discovered just recently, and the function of this protein wasn't really known until maybe 1 or 2 years ago, which is recently, in scientific research. And this protein actually drives the formation of the axon, and that's the projection that leaves the cell body and goes out to the skeletal muscle. And in normal development and in the normal system, if axons do not progress far enough to reach their target, they are going to be eliminated. So it is likely that this mutation in the protein does not allow the axon to grow to where it needs to go, and so you're going to get the killing off of that cell. Now, the inability for proteins to really function properly, you can look at different ways of trying to address the issue, but the SOD1 protein is really, really good because it's been around for a long time, and we're really familiar with what it does. And SOD1, when it becomes misfolded, results in a lot of oxidative stress in the cell. And this, in turn, is going to kill one of the most important organelles that is within that cell. And that organelle is called the mitochondria. Now, what the mitochondria do is they are essentially the little organelles. They're the powerhouses that take the glucose and the fat from the body and turn it into energy. And that energy sustains the life of any cell that mitochondria is sitting within. Now, what we know in MND is that there are a lot of defects in mitochondria seen in the skeletal muscle and in the neurons. So if you have a look at these two images, and I'm not sure if you can see that one very well, but this is an image taken from a normal individual. And you can see that the mitochondria are quite nice and big, but in an MND patient, some of these mitochondria are quite small, and you can see that this one's actually rupturing, and all of its contents are spilling out into the cell. And over time, when you get this constant loss of mitochondria, and the mitochondria are dying and they're being, mopped up by all the immune sort of markers in the cell, what you get is an eventual energy deficit. And these cells can't survive because they don't have the energy that they need to survive. Now, the other interesting thing about mitochondria and energy deficit is that the activity of a cell is also dependent on its metabolic state. And what we do know is that in these neurons in MND, these cells are considered to be hyperexcitable. So they have a lot more activity than what they should do. And this has really been exemplified recently in motor neurons that have actually been derived from MND patient skin cells. So they take the skin cells, they turn them into stem cells, and then they force those to become motor neurons. And you can see that in motor neurons that have been derived from control patients and motor neurons that have been derived from MND patients, there is a difference, and I hope you can see in the number of spikes that you can see in these recordings. And these spikes are really an indication of the cell activity. So the more spikes you have, the more crazy the cell is really sending signals around the body and its connections. And when cells send signals, what they do is they do it through a chemical called glutamate. And glutamate the signal gets propagated down the first cell, and glutamate gets dumped into this space, this synaptic cleft, which is an area that's between the first cell and the receiving cell. When the receiving cell takes up the glutamate, it sends the signal on to the next target. Because glutamate can be quite toxic when it's present at very, very high levels, there are these other cells in this environment that express these special proteins that basically mop up any excess glutamate that's in that area. And what you see in MND is that with the cells spiking and having a lot of activity, there's this flooding of glutamate into that space. And these astrocytes don't have enough of these proteins to mop up that excess glutamate. And so you're just getting this constant dumping of glutamate into the system, no cleaning up of this glutamate, and that will eventually kill off the cell. What we are doing up in Brisbane at the moment now, and with a talented young man who's just finished his PhD, is we're trying to figure out whether or not the activity of the cell and the amount of glutamate that's being released into the system can be dictated by the way that the cell looks, the morphology of the cell. And what we've done is we've taken some mice that are really, really young. They haven't developed any symptoms. And in the normal mouse that's 21 days old, you can see this beautiful cell here, and it sends out this really long projection out into the distance. And there are some nice little projections closer to the body. And this long projection will send signals down to the next cell, whereas these projections will receive signals from incoming cells. And what you see in the mouse that has the mutated gene for MND and will eventually develop MND later on in its life, this mouse, when it's pre-symptomatic, that long projection is a lot shorter than what you see in the normal mouse. And the number of these short projections closer to the body, there are also less of them. And so what we have to think is this morphology of the cell really dictating how it's connecting to other areas in the body? And because these cells are shorter, does that make their physiology different such that they are susceptible to being killed off over a normal cell that's functioning properly? And does this change the way in which the neuromotor system is formed in MND? So now that we've sort of gone over the genetics and the molecular and cellular mechanisms that underlie disease, what we really have to think about is there anything else that can influence our genetics and our cellular and molecular physiology? And two things I briefly want to touch upon are about the environment. Now in 2013, Rachel Dunlop in New South Wales really started to look at this work on the blue-green algae. So what happened was there was this population of people in Guam who had a very, very high incidence of MND. And they thought that maybe it was just something in the environment. And it turns out that the people in Guam were eating this delicacy called bat soup. And it's a whole bat that sits in the soup. And it's not necessarily the consumption of the bat itself that would lead to the susceptibility to MND, but it was really what the bats were consuming off the trees, the fruits of the tree that they were eating. And at the roots of these trees, there was a large amount of blue-green algae. And it turns out that what the blue-green algae does is it secretes this toxin that is very similar to the amino acids that naturally occur in our body that are important for building proteins. So the belief is that when people ingest this toxin, it secretes these toxins, and the body cannot recognize or distinguish the difference between the toxin and the amino acid, and it incorporates that toxin into its protein formation process. And that basically gives you the production of a non-functional protein. The other thing in the environment that might affect MND, or the onset of MND, which is a little more controversial, is this idea of elite athleticism. And this was brought up in the ALS meeting last year because it was based in Milan, and there is this reported high incidence of MND in Italian soccer players. So it's of a significant interest to the Italians to look at the relationship between sport and MND. And of course, with sporting heroes like Lou Gehrig, I don't know if you know Steve Gleason. He's an American football player who developed MND, and Joos van der Westhuizen, which is a Springbok rugby union player, and more recently Peter Frates, who all you know, started the ALS Ice Bucket Challenge phenomenon. There is anecdotal evidence that elite athletes develop MND, but whether that's just because they're famous and everyone knows about them, that it sheds more light on this athleticism, really is a bit of a topic for discussion. And so a direct link between increased physical activity and the development of MND really remains to be determined. Okay, so if we know that there are misfolding proteins and glutamate toxicity and mitochondrial dysfunction that causes disease, and we know that there's electrical excitability, and we know that the environment can affect the disease, have we been able to do anything to take this knowledge to modify disease in patients? And the answer is kind of, but it's not really to do with the classical things that we've been researching. Really, it's about how fat a person is. And the idea of body mass index predicting disease outcome really came about in 2010. And it was followed up in 2011 by this group, Paganoni and colleagues. And they showed that MND patients who had a BMI between 25 to 35 had improved survival compared to people who were very slim or compared to people who are quite large. And what's more interesting is that if you have a higher pre-diagnostic BMI, that's associated with a lower rate of MND and a decreased risk of mortality. So all of this about body fatness works off the premise that there is altered metabolic balance in MND. And a lot of research has been done in France that shows that MND patients tend to have a higher resting metabolic rate. And under normal circumstances, the energy demands of the body are met through food intake, and anything that's not consumed or used as energy is stored in the liver or the fat. And what happens in MND is if you have increased metabolism and you have decreased food intake, you have an increased dependence on your stored energy. And we do tend to see a lot of weight loss in MND patients. Sometimes it's very rapid, and sometimes it's very, very slow. I mean, what we have to understand is this weight loss is not necessarily just attributed to the loss of muscle mass, because you will see that a lot of patients also lose fat mass. Now, the idea underlying this benefit of fatness in MND comes from this: as disease progresses, you have this increase in metabolic pressure on the body. And as you burn through all your energy stores, you've reached a point of catastrophic failure to accommodate demand. And that's when the patients start to progress very, very quickly, because there is nothing left for them to burn through. And, when we look at this and people think, oh, it's just as simple as feeding someone more. Can that really work? It seems so straightforward. A study that was recently published early this year, a phase 2 clinical trial, shows that patients who are quite far progressed in the disease, all of the patients in this study were fed by a PEG. And those who received high-calorie, high-carbohydrate supplementation through that PEG, or high-fat, high-calorie supplementation through that PEG, had better outcome. Those patients who didn't receive any supplementation saw 3 of the patients pass away in that study cohort. And so what this really, really leads us to think is that maybe by changing the way in which the body is responding to the disease, we can actually modify the course of disease and extend survival. And a lot of people are now looking at dietary interventions and the use of, I don't know if Leonie is here anymore, of supplements like vitamins and minerals. And because we don't really know what they do, a lot of research needs to be done. And I've actually set up a collaboration with someone in Chile who does a lot of work on nutrition and nutraceuticals. And so she'll be doing research with me, hopefully next year, to look at how these nutraceuticals will affect disease progression. So that was my answer to one question. Okay. So if we're able to modify the course of disease, that's really great. But how do we track the fact that we're changing the course of disease? Okay. And two things that are really coming to light is the use of blood biomarkers and imaging. So this is an image of a neuron and its long projections. And what is contained in these projections is a protein called neurofilament. And when axons start to degrade, what happens is the neurofilament is shed out into the bloodstream. And what people have done is they've tried to use the levels of phosphoneurofilament in the blood or in the plasma to see whether or not you can, it's an indicator of how quickly the disease progresses. And in this study, and I won't try and pronounce that name because I can't, high levels of neurofilament are associated with a shorter disease duration. And this data of phosphoneurofilament is really something that we see up in Royal Brisbane in our cohort of patients. And so I believe that the neurofilament is really going to be a promising biomarker, at least for how the axons are degenerating throughout the disease process. And the other thing that's really moving forward, and everyone loves it because it produces pretty images, is this imaging technique of MRI and DTI and all these things. So what you can do with imaging is you can look at the functional connectivity in patients non-invasively. You just image them and you can see whether or not connections in the brain are making the same connections in the brain as what you would see in a normal person, and whether connections from the brain down to the spinal cord are degenerating. And this study really shows that the cortical thickness in MND patients is a lot smaller than what you see in a normal control, and this could probably be attributed to the loss of the cells in the brain. And what they also see, there is a decrease in functional connectivity. So basically, these long projections that connect to each other from the brain to the spinal cord are being lost in patients. And you can really track this non-invasively through the imaging process. And the imaging is at the stage now where we could initially see a few cells quite well, down to being able to image cells down to a single resolution in mice with a 16.4 Tesla, which is one of the most whiz-bang imaging machines that I really want to use. Okay, so how far have we come in treating MND? And I know that I said that since Riluzole we haven't really come up with much, but this is really not to say that there aren't any clinical trials going on. So I jumped onto the ALS TDI website and I was quite impressed at the number of clinical trials that are actually going on at the moment. And what I hope you can appreciate is that the clinical trials that are further on, the phase 2, phase 3, are clinical trials that are aimed at addressing these molecular and cellular mechanisms that underlie MND that we've known about for a really long time. But these earlier these trials that are phase 1 or that haven't even been given a phase status yet, they're really looking at the effect of exercise and nutritional supplementation. So the clinical trials that are coming through are really a few steps behind what we are discovering with our scientific research. Okay? And the other thing I want to touch upon when it comes to trials is the use of stem cells. There are a few stem cell trials going on, and they're due to be finished this year or next year. But I would have to say that based on the data that we have, stem cell therapy is really in its infancy. And this is really demonstrated in this paper by Glass and colleagues in 2012, where they took these fetal stem cells and transplanted them into 12 MND patients. And the newspapers really got onto this and said that stem cell therapy really saves MND patients and extends life. But when you pull out the scientific paper and you have a look at what's been done, what you can see is when you look at the functional rating scale and the lung capacity of these patients who are receiving the treatment, they really like to harp on about patient 11, who's the black line here. And they do have a bit of an improvement in the functional rating scale and a modest improvement in their lung capacity. but the 11 other patients don't really show much. And in fact, when you look at the results in this study, 11 patients had adverse events, and this was associated with a surgery or a heart attack or whatnot. Okay, so I think there's a bit of a way to go with stem cell therapy. I'm not saying that it's not going to be an option in the future, but I think at the moment it needs a lot of development. But if stem cell therapies aren't going to work for us, what can we learn from the stem cells? And the group in Harvard, the Harvard Stem Cell Institute, they are really leading the way when it comes to stem cell research in MND. And what these guys have been able to do is they've taken skin cells from a normal person or from a patient who has a SOD1 mutation, and they take these cells and turn them into motor neurons. And you can see here that the motor neurons look very, very similar. But what they also show is that these motor neurons have this hyperexcitability. They're just firing like crazy, just like we see in our mice. And so although Rob has said that our mouse models are not necessarily the best, and I would agree that they're not the best, these cells from the patients have a similar hyperexcitability that we see in the mice. So this is not to say that we should get rid of the mouse, as cute as they are. There is something that we still can learn from the mice when it comes to the mechanisms of disease. And what's really interesting about these stem cells now is that they're from the patient. And what we can do is take these stem cells and start looking at other treatments that are available on the market. And that's what people have started to do. And they've looked at these potassium channel openers. And the reason why they've looked at these potassium channel openers is because potassium channels play a very important role in regulating the activity of the cell. If the cell is firing constantly, these potassium channels are meant to open and stop that from happening and return the activity to normal. So what these guys in the US did, they decided, well, you know, this anti-epileptic sort of works by opening these potassium channels. So let's just put it on these cells and see what happens. So you've got these hyperexcitable motor neurons here, and when you put on this retigabine, you normalize the cell activity. And not only is the cell activity normalized in these neurons, they're also able to extend the survival of these neurons in the dish. And they live much longer than the neurons that aren't treated with this potassium channel opener. And the drug is quite effective at a very low dose, but we have to remember that this is in the dish. What this group is pushing to do now is to try and move the use of retigabine into clinical trials for MND, because we have already overcome that issue with safety and tolerability, because it is being used as an anticonvulsant in epilepsy today. The other drug that might be of interest is diazoxide. Diazoxide is used to treat hypoglycemic states and hyperinsulinemia. And there was a patent filed a few years ago that want to use diazoxide as a treatment therapy in MND. And the reason for this is because of this graph that's presented in the patent, which you can find on this website here, is that animals that are treated with diazoxide, which is similar to what people are getting clinically, can extend survival when compared to mice that aren't given diazoxide. So it would seem that, much like riluzole, which strangely enough normalizes cell activity, it would seem that the drugs that normalize cell activity might be the way of the future to go for the best possible drugs that are immediately available to us. So what does all of this research and knowledge mean for the future? And Jeanette's going to kill me because this map is incorrect, but, because Royal Brisbane is not part of University of Queensland, but we're all friends. The growing group of researchers in Australia, and I think maybe when Jeanette first made this map, there weren't that many people listed on this map, I'm not sure, but this growing number of researchers in Australia is really showing that we're not too bad when it comes to MND research. We are making a lot of discoveries, and there is a lot of collaboration between us. We work with groups in Sydney, we work with groups in Melbourne. Hopefully we can work with groups in Perth and Tassie soon. And even Brisbane alone, we have a lot of global connections. And I was saying we've now hooked up with someone in Chile. We're doing some stem cell research in China, and we're working with people in the US and Europe for looking at how we can treat disease and find the underlying causes, and South Africa. And I know there's not much research in South Africa, but the people in South Africa are actually really important to us because they don't get very much care when it comes to MND. And by communicating with a few patients I know in South Africa, I can gain a lot of information about what we should be doing and where we should be moving with our research when it comes to what patients who aren't very well looked after really want. So, you know, the research is really going in a few different directions. We're trying to figure out what the causes are that underlie disease, and that's from genetics to protein to energy, function in the environment. And we're tracking how disease is progressing, and these biomarkers are really, really promising. We're looking at how we can modify the course of disease, how we can translate what we see in mice into the human now with the power of being able to derive these motor neurons from patient cells. And we're trying to collaborate globally to be able to bring all these expertise in together to do all of this research. So on this note, what I'd like to do is draw your attention to some insight of a very famous physiologist, and he's considered maybe the best man in science, really. And the red box moved. Okay, so what I wanted to focus your attention on was this part here. All of the vital mechanisms, however varied they may be, have always one goal: to maintain the uniformity of the conditions of life in the internal environment. And we know that in MND, it is these varied mechanisms, and a lot of them, that are not maintaining this internal environment. And in order to be able to treat MND, we cannot just focus on one aspect of the disease. We have to be able to incorporate all of the knowledge that we know across the mechanisms, to the biomarkers and the environment, and to what we see in human cells and mouse cells, to be able to move forward to developing a world without MND. Okay, so thank you.